Hey bio team. All the cells that you guys have seen so far, whether it's prokaryotic cells like this bacterial cell or eukaryotic cells like this plant cell, all have at least two organelles in common. They all have a cell membrane, and they all have ribosomes. And the reason that cells need ribosomes is because ribosomes build proteins, which is what the cells themselves are built from. So for example, these red blood cells have inside them hemoglobin proteins, which help them carry oxygen to different parts of the body. As for what a protein itself is built from, if you were to zoom in on a protein, you'd see that it's this long strand of smaller pieces bonded together and then folded up into a very specific shape. And these smaller pieces that get bonded together are called amino acids. And their name is pretty straightforward. An amino acid is made of an amine group, which has nitrogen in it, and an acid group, which are bonded together by a central carbon atom. Now, in order to form a protein, these amino acids need to bond together. So we'll watch that happen here. And what you'll notice happening is that the hydrogen on the amine group from one amino acid and the hydroxide on the carboxyl group from the other amino acid broke off to form a water molecule. And in the process, the carbon and nitrogen bond together in what's called a peptide bond. Now, this process is also called a dehydration or a condensation reaction, because water molecules are being broken off each time an amino acid gets bonded onto this chain, onto this protein. As for which protein actually gets built, that's dependent on these R groups that are attached to the amino acid. And each type of amino acid has its own unique R group. And you guys have actually seen these before. Last year when you guys were going through codon charts, you can see the different types of amino acids. Each type of amino acid it has a different R group as its side chain. Now, the type and order of amino acids will determine the overall shape of the protein. And that has to do with the charges on the different amino acids. Some amino acids have R groups with slightly negative charges. Some amino acids have R groups with slightly positive charges. And so as a result, these opposite charges will attract together, and the chain will actually bend and fold back in on itself. We'll watch this happen here. These are often referred to as like clover leaves, or the chain will kink. But regardless, it's the attraction between these amino acids that folds the protein together and holds it into a specific shape. And so one way biologists will say this is they'll say that a protein's primary structure, that is the order of its amino acids, will determine its secondary structure, that is how the amino acids fold together. And so to go back to our example of blood cells, in a healthy blood cell, there's a normal amino acid sequence in which protein folds up normally. However, if someone has sickle cell anemia, their amino acid sequence has been changed as a result of a mutation. And as a result, the proteins fold up in a different way. They're misshapen, and that different shape causes this sickling in the cell. Which brings us to one of the key ideas of proteins, and that's that a protein's shape is necessary to its identity. It's necessary to its function. So for example, this enzyme protein, lactase, is made of this long chain of amino acids which have folded together into the perfect shape to break apart the substrate lactose, which is the sugar in milk. And so we can watch that happen here. And it's possible that if there were even a small change in this protein's primary structure, if we were to switch some key amino acids, a few changes could result in the protein denaturing or changing shape, folding up in a different way and inhibiting this protein from working, preventing the protein from breaking apart the substrate. And so that's it. You guys have a couple of practice questions. We'll see you in class next time.